I am Professor S. Rao, HOD Department of Botany, GN College, Dhurwa. Today I am going to teach the students of semester 3rd, BSc Botany Honors, paper 7. The topic of today's discussion is Mendelism. So, before going into this Mendelism, I would like to I would like to draw your attention toward the basics of genetics. So, Mendel is rightly called as father of genetics. Then, what is genetics? Genetics is defined as the branch of biology that deals with the study of heredity and variation. Again, we have two terms, that is heredity and variation. So, now I define heredity in this way: the inheritance or transmission of characters from one generation to the next generation is called heredity. Now, what does it mean by one generation and what does it mean by next generation? By saying one generation means parents, and by saying next generation means their progeny, their next generation. So, again, heredity can be defined in this way: the inheritance of characters from parents to their offspring is called heredity. Here, parents means father, mother, or male, female, and offspring means their progeny, their next generation, means son or daughter, male or female. This is called heredity. So, heredity is nothing but inheritance of characters from parents to their offspring is called heredity. Now, the second important term of this genetics is variation. It is differences existing among the individuals of same species and also among the offspring of the same parents are termed as variation. Now about Mendel. Who was Mendel and why Mendel selected Physamphytum, that is pea plant, as his experimental materials? So these two aspects are very important to know about Mendelism. So Mendel is called as father of genetics. He was the first person to propose the law of inheritance. And why did Mendel select garden pea plant as his experimental material? There are so many reasons behind taking garden pea, that is Pisum cytbum, as his experimental material is that: number one, pea plant is self-pollinated plant. Pea plant is self-pollinated plant. Now, reason 2, pea plant shows 7 varieties of characters, that is seed shape, round and wrinkled, seed color, yellow green, flower color, purple and white, pod shape, that is full and constricted, pod color green and yellow, flower position axial or terminal, or stem height tall and dwarf. So, as we have seen, there are 7 characters found in the garden pea. Therefore, Mendel selected pea plant as his experimental material. These are the reasons for taking the garden pea as Mendel's experimental material. Now, about phenotypic character and genotypic character. Phenotypic characters means external expression, external appearance of color, height, whatever it may be, through eyes is rightly called as phenotypic characters. And what is genotype? What is genotypic character? Genotypic character is internal constituent of an organism. That is, it is the internal genetic makeup for its color, its height, its shape, whatever it may be. So, it is the internal factor, and the phenotypic character is external appearance. So these two words, phenotypic characters and genotypic characters, is commonly used in the field of genetics. Now, the Mendel performed his experiment in pea plant by taking these 7 characters. He took seed shape. When Mendel crossed round shape seed with wrinkle seed in F1 generation, he got all were round, and in second generation, that is F2 generation, 1 is to 2, 1 is to 2 is to 1, that is 1 was pure round, 2 were impure round, and 1 was pure wrinkle. Similarly, when he took seed color, he crossed yellow seed color with green seed color. In F1 all are yellow, and when he crossed the offspring of the F1 in second filial generation, he got again 1 is to 2 is to 1, 1 pure yellow, 2 impure yellow, and 1 pure green. When he performed his experiment with flower color, he took violet color and white color. When violet color and white color crossed in F1, all are violet color, and when he crossed the product of F1 in second and F2 filial generation, he got 1 is to 2 is to 1, 1 is pure violet, 2 is impure violet, and 1 is pure white. Similarly, in pot shape, he took full pot shape and constricted pot shape. He crossed full pot shape with constricted pot shape in F1. All are full pot shape. But when the progeny of this F1 were crossed in F2, he got again 1 is to 2 is to 1. 1 is pure full and 2 were impure full and 1 was pure constricted. Similarly, the pot color, green pot color, when crossed with yellow pot color, the F1 are all are green, all are green. But in F2 again 1 is to 2 is to 1, 1 was pure green, 2 were impure green, and 1 was pure yellow. When he crossed the flower position, axial flower position was crossed with the terminal flower position, the F1 all are axial, and when the offspring of F1 were crossed together in F2, that is next generation, he got again 1 is to 2 is to 1, 1 was pure axial, 2 were impure axial, and 1 was pure terminal. And last one, he when he crossed stem height, that is tall plant was crossed with dwarf plant, again F1 all are tall. But when the offspring of F1 were crossed together in F2, he got again the same result, that is 1 is to 2 is to 1. 1 was pure tall, 2 were impure tall, and 1 was pure dwarf. So Mendel performed his experiment in all these 7 characters, and he got the same result. In F1 all are dominant, and in F2 1 is to 2 is to 1. So again I would like to repeat what were the 7 contrasting traits Mendel performed his experiment in his the field of genetics. Character he took were stem height, and its contrasting traits is tall and dwarf. Flower color, its contrasting color traits is violet and white. Flower position, its contrasting traits is axial and terminal. Pod shape, its contrasting traits is inflated and constricted. Character 7 pod color, its contrasting traits is green and yellow. Seed shape, its contrasting traits is round and wrinkled. And seed color, its contrasting traits is yellow and green. So these all are 7 characters and their contrasting traits taken by Mendel during his experiment.